Monoclonal antibodies are antibodies with the same tertiary structure and antibodies which are produced from identical plasma cells. You need to know the uses of monoclonal antibodies, and those are for the treatment of diseases such as cancer by targeting drugs to those cancer cells, or for medical diagnosis. This is an image to help you visualize how monoclonal antibodies can be used as a treatment option. In this scenario, the monoclonal antibodies are the red Y-shaped structures, and attached to those are an anti-cancer drug. As the antibody has a specific tertiary structure, it will bind onto a complementary antigen on the surface of cells, such as breast cancer cells, carrying the drug to that cell. Another use of monoclonal antibodies is for medical diagnosis. Monoclonal antibodies can be used in the ELISA test, which is used to test for the presence of pathogens such as HIV. There are different types of ELISA tests, such as the direct and indirect ELISA test. You do not need to know the differences between these two types and can describe any type in an exam. In the direct ELISA test, the antibodies complementary to the antigens are stuck to the bottom of a well plate. A second solution containing a second antibody complementary to the antigen is then added. This antibody contains an enzyme which can cause a color change if the antigen was present. In the indirect ELISA test, the order of events are slightly different. Firstly, the antigens are attached to the bottom of the well. This is followed by addition of the first and then the second antibody with the enzyme attached. Before adding the second antibody in the direct or indirect ELISA test, the surface must be washed to remove any unbound antigens or antibodies.